With very warm greetings. This is Ali Farhadi, Associate Professor of Medical Virology, working as a scientist at the Department of Medical Biotechnology at Shiraz University of Medical Sciences in Iran. I have been researching on protein therapy for the last 8 years, and during my research, I discovered a compound called pegylated TAT-F7TA, an anti-arrhythmic agent with favorable effect on heart failure caused by arrhythmia. I extensively focused on this compound to evaluate its efficacy by in silico as well as experimental tools, including in vitro and in vivo experiments.
According to the WHO, cardiovascular diseases called CVD are the number one cause of death worldwide, representing 31% of all global deaths and 18.6 million lives per year. Common cardiovascular diseases include heart failure, cardiac arrhythmia, coronary artery syndrome, stroke, hypertensive heart disease, cardiomyopathy, and etc., whose pathogenesis differs from each other. CVD's burdens are predicted by WHO to stay at the first rank until 2030 due to the aging of the population and the increasing prevalence of diabetes and obesity. Despite this enormous importance for public health, existing drugs for heart failure and cardiac arrhythmia are still far from being ideal, as they display perilous side effects and cannot be administered over extended time periods. Especially, cardiac arrhythmias are difficult to treat due to major side effects of common antiarrhythmic drugs. However, due to their effects on the cardiac action potential and thus cardiac conduction speed, they are prone to proarrhythmic side effects. Therefore, novel therapeutic strategies that suppress the initiation of arrhythmogenic signals inside cardiomyocytes are currently in focus of the research for novel and safer antiarrhythmic therapies. Here, the drug invented due to its components as a powerful agent is an innovative way to treat all types of arrhythmia and in a completely targeted way. Also, this drug do not show cardiac toxicity and do not cause noticeable pathology. This protein has shown a remarkable and very promising effect, which has been confirmed by many in vitro and in vivo laboratory tests. The components of this protein include TAT, F7, TA, and polyethylene glycol. TAT is a transactivator of transcription derived from HIV-1 and showed effective transduction capabilities for various cargoes. It consists of positively charged transduction through negatively charged phospholipid on cell membrane. F7 is a novel compound binds to the outer mitochondrial membrane VDAC2, enhances mitochondrial calcium uptake, and restores rhythmic cardiac contractions in vitro and in vivo experiments. Tannic acid has appealing properties as a molecule because of its strong affinity with biomolecules including DNA and proline-rich proteins such as thrombin, gelatin, collagen, and mucin. Protein and peptide drugs hold great promise as therapeutic agents. However, many are degraded by proteolytic enzymes, can be rapidly cleared by the kidneys, generate neutralizing antibodies, and have a short circulating half-life. Pegylation, the process by which polyethylene glycol chains are attached to protein and peptide drugs, can overcome these and other shortcomings. The present invention consists of three phases of study. The first step was in silico phase. In this phase, three components of this drug, including TAT, F7, and TA, were examined using these databases and softwares. And finally, the design of this drug was successfully performed by the obtained data. The second phase of the study was in vitro phase. At first, TAT peptide and F7 were synthesized. In the next step, TAT peptide was conjugated to F7. And then tannic acid was pegylated using polyethylene glycol. And finally, TAT F7 was attached to pegylated tannic acid. The accuracy of pegylated TAT F7TA was confirmed by the following methods, including zeta potential, scanning electron microscopy, and etc. And also, its solubility and stability was fully evaluated. On the other hand, the function of pegylated TAT F7TA was evaluated. The tests performed to analyze the effect of this compound are as follows. In this image, the specific penetration and delivery of pegylated TAT F7TA into the H9C2 heart cells is shown. In this slide, the effects of pegylated TAT F7TA agent in vitro and in vivo experiments are shown. Figure A represents the increased mitochondrial calcium uptake in H9C2 permeabilized cardiomyocytes treated by the compound. A significant increase in mitochondrial calcium uptake was observed in cells treated with pegylated TAT F7TA agent. Figure B represents that the compound reduces diastolic calcium waves in cardiomyocytes of mice models. After the addition of ISO, an increase in the number of calcium waves per minute was observed, which was significantly reduced after simultaneous treatment with ISO and the pegylated TAT F7TA agent. Figure C represents patch clamp recordings of mice cardiomyocytes showing that spontaneous action potentials, which were significantly reduced by pegylated TAT F7TA, after the addition of ISO, an increase in diastolic action potential was observed, which was significantly reduced after the addition of the compound. Figure D represents the results of the ineffectiveness of the pegylated TAT F7TA agent on CAMP accumulation in freshly isolated hearts of mice models. While the addition of ISO caused a significant increase in cellular CAMP, the pegylated TAT F7TA agent had no effect on the ISO-induced increase in cellular CAMP levels. As the conclusion, pegylated TAT F7TA compound acts as an anti-arrhythmic agent due to the presence of F7 and even improves the heart failure caused by arrhythmia, which is delivered specifically to the heart tissue due to the presence of tannic acid, and penetrates into the heart cells and mitochondrial membrane due to the presence of the TAT peptide. By targeted binding to VADC2 protein, it can be used effectively in arrhythmia and heart failure caused by it. Also, by pegylation of this compound, in order to increase half-life, stability, and reduce renal clearance, we are able to greatly reduce the common limitations associated with the use of therapeutic agents and peptides. Finally, the conjugated TAT F7TA agent can offer a more specific and efficient function by removing the limitations on conventional treatment methods.